Welcome to the second installment of our video lectures on inheritance. This time we are focused on pedigrees and variation. Our guiding questions continue to be what patterns of inheritance exist in plants and animals, and what is the molecular basis of inheritance patterns. Our objectives for today, we're going to analyze lots and lots of pedigrees, all of these different modes of inheritance. We're going to also look at variation in phenotypes, distinguishing between continuous variation and discrete variation. We're going to talk about what even is polygenic inheritance, and then we're going to take a quick look at some data analysis and how we can use box and whisker plots to evaluate continuous variation. And here we have our first example of a pedigree. Pedigrees are charts that are records of an organism's ancestry. They also show how traits can be passed through generations. Let's say I'm looking at this individual here. This individual has a square. We use squares to denote people with testicles, males. We have circles to denote people who do not have testicles, people who have ovaries instead, females. We color in the circles and the squares if people are affected with the disorder. We do not color in the squares and the circles if people do not have the trait or the disorder. This particular pedigree is showing us the inheritance of a disorder known as Huntington's disease. It is a disease that affects humans. It is a neurodegenerative disease. That means that the brain kind of breaks down. It is fatal. It is an awful disease to watch happen to a parent. What's crazy about this one is that it isn't fatal until later in life. And so people do not start to show symptoms until they're maybe in their 40s, which means that they've already had their children and passed on this awful disease before they realize that they themselves have it. So pretty rough disease. It is interesting because it is a dominant trait. Most of our fatal disorders are not dominant. They're mostly recessive. That's how they kind of hide in the population. This guy is fatal and dominant because that fatality doesn't occur until later in life. Again, that trait has been passed on before the people realize that they are symptomatic, before they realize that they have the disorder. It is also autosomal. Whoa, if I could spell autosomal, that means that it's on an autosomal chromosome, not the X chromosome, not the X chromosome, one of those first 1 through 22 chromosomes instead. We know this by evaluating the pedigree because there are equal numbers of males and females with the disorder. Remember that those sex-linked traits are going to show a higher prevalence in males than in females. Let's analyze this pedigree. It's a little bit different from the last one. Notice that we have a pair of individuals here, female parent, male parent. The male parent is affected, the female parent is not. They have offspring. None of their offspring are affected. But when this unaffected male has children with this unaffected female, notice neither of these parents are affected. They do have an affected offspring. This tells us that this trait must be recessive. The recessive allele was masked in these individuals, in these parents, by the dominant allele, but they must have been carriers in order to give it to that offspring. It must also be autosomal. We know that by analyzing the pedigree, because there are equalish numbers of males and females who are affected. If it were sex-linked, we would have way more squares colored in than we have circles colored in. An example of an autosomal recessive trait in us humans is sickle cell anemia. So let's say that this is a pedigree showing sickle cell trait popping up in individuals in a family. We use H's for sickle cell trait because it's a mutation on the hemoglobin protein. We use capital B for normal hemoglobin and lowercase b superscripts to show that mutation that leads to sickle cell anemia, that clumpy hemoglobin that causes the red blood cells to get curvy and then stick on each other. So this individual here must be H little b must be homozygous recessive in order to have sickle cell anemia. The two parents that we were looking at here, their genotypes must have at least one of those H little b's, but since they do not have sickle cell anemia, they must both be heterozygous. So they must have one of those dominant alleles that masks that recessive sickle cell allele. This individual also H big B, H little b. Where did this individual get that H little b? From this parent. This one must be homozygous recessive also in order to have that sickle cell trait. This individual we don't actually know is going to be either HB, H, another big B, or could be heterozygous. We can't really tell from this set of offspring. These guys, though, I definitely know that both of them are heterozygous. H big B, H little b. H big B, H, little b. And we know this because they have children who are affected. These two individuals must have H little b in order to have that sickle cell trait. The other two offspring, we can't tell because when we do that Punnett square, HB, 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 big B's and little b's, we could have an offspring that is homozygous dominant. We could also have offspring that are heterozygous carriers. And then, of course, there is the potential to have offspring that are homozygous recessive that have that sickle cell trait. Be careful with the Punnett squares. This does not mean that one child is homozygous dominant and two children are heterozygous and one child is homozygous recessive, because notice in these four children, definitely two of them have that H little b genotype. Our Punnett squares just tell us that there is a 1 in 4 chance of being homozygous dominant, a 1 in 4 chance of being homozygous recessive, and a 2 in 4 or 1 out of 2 chance of being heterozygous. It doesn't tell us that you're going to have exactly 4 children and each of them has exactly that genotype. Feeling good? All right, let's look at another pedigree with a difference inheritance pattern. So this pedigree is showing us incomplete dominance, and the grade versions are those blended phenotypes. We talked about 4 o'clock flowers. Here they are, where red and white, neither one of them is completely dominant over the other. Instead, there is incomplete dominant, where the heterozygotes, rather than being red or white, are a blend of red and white. They're pink. And so here, let's say that this is the red flower, and so this is going to be CRCR. This grade version is the intermediate CRCW pink. And when these two guys cross, we've got CRCR and CRCW in our Punnett square, and we have oh so many C's, and we have some R's. And we have some W's, and we have some more R's, and we have some R's and some W's. So we should have about half of the offspring red and half of the offspring pink. And look, we have some reds and we have some pinks. But if we cross this pink with a white, now we still have lots of C's, lots of C's. But here we have an R and a W to make the pink, and we have W and W to make the white flowers. This parent is this parent. This parent is this parent. And we have, again, so many C's, and we have R's and W's and R's and W's and W's and W's and W's and W's. Wait, what happened? We should have half pink and half white, but we have all white. Again, remember that these Punnett squares do not tell us exactly the outcomes of the offspring, just the probability. This guy had a 50-50 chance of being pink, a 50-50 chance of being white. It got white. This one also had a 50-50 chance of being pink or white, and it also got white because it had a half chance of being white. So incomplete dominance here we can show with some intermediate shading. Instead of just having black and white, we have now some grays in the middle. Sex-linked traits can also be shown in our pedigrees. This one is a pedigree for colorblindness in human. It is a sex-linked trait. That means it is on the X chromosome. Remember that these traits that are on the X chromosome, females have 2 X's, males have X's and Y's. So females could be homozygous dominant, females could be heterozygous, females could be homozygous recessive. Males cannot be homozygous or heterozygous because they only have one. And so they are what we call hemizygous. Hemizygous, they only have half of the genotype. They can be hemizygous dominant or hemizygous recessive. These half circles are showing us our carriers. So these are the heterozygous females. So here we have a carrier female and an unaffected male. So neither of these people are colorblind, but they have children, and one of their female children is also a carrier. The male child has colorblindness. Let's look at that in a Punnett square. So that mom here, who is a carrier for colorblindness, I'm going to give her a capital C and a lowercase c because she is a carrier. So she has normal vision. Because of that dominant allele, she is a carrier of colorblindness. And then dad is XY, not colorblind, not affected, and so he has also a capital C for normal vision. When we do the Punnett square, we see possible outcomes, possible genotypes for children. So they could have a female child who is not a carrier. They could have a female child who is a carrier. They could have a male child. Whoa, that was not what I meant to draw. They could have a male child, that should be a capital C. With normal vision, they could have a male child who is colorblind. This male child happened to get that spot on the Punnett square. There was a 1 in 4 chance of a child being born who was male and colorblind. This individual then, their genotype is little cy. This individual is going to be capital C, capital C. Notice that their children, their male children, neither of them are colorblind. Let's look at this interesting Punnett square. So we have mom is big C, big C. Dad is little cy. So we could have female children who are big C, little c, big C, little c. We could have male children who are big C, nothing on the Y, big C, nothing on the Y. This means that none of their children should be colorblind. Their female children are going to all be carriers. Their male children will have totally normal vision. This is what happened with me. This is me and my children, and my ex is colorblind. None of my children are colorblind, but my female children, my children with ovaries, are all carriers for colorblindness. Super interesting, right? So interesting. Here is a pedigree for another sex-linked trait. This one now is hemophilia. Hemophilia is another disorder on the X chromosome. It is a blood clotting disorder. People get little cuts and they cannot stop bleeding. This happens to be royal families of Europe. There is a lot of hemophilia in the royal families of Europe, and let me show you why. So Victoria and Albert, they had so many children. This child and this child, they're sisters. They had children. Their children had children. So these are first cousins. The royal family thought that they were maintaining the purity of their bloodline. It was a little incestuous. There was some inbreeding in the royal family, which led to lots and lots of hemophilia popping up because they just kind of kept that allele rolling around within their bloodlines. Here we have that the Romanovs, so that last royal family of Russia, lots and lots of hemophilia there. If you've ever seen the musical Anastasia, it's pretty good. You might be thinking, well, that inbreeding thing, that happened a long time ago. It doesn't happen anymore. Elizabeth and Philip just recently passed away. Here is King Charles of England, his parents, Queen Elizabeth, Prince Philip. Let's look at them. Here's their parents. Here's their grandparents. Here's her parents. Here are their grandparents. Their great-grandparents were siblings. Their great-grandparents were siblings. So yeah, it used to happen, but not really that very long ago. Crazy sauce, right? So all of these different traits can have different kinds of variation. We can have continuous variation, we can have discrete variation. Continuous variation, like this lovely normal curve that we have, where we have the most frequent being something that's kind of average, something that's very extreme is going to be very less frequent. So an example of this would be human height. We have a very few people who are very, very, very short. We have a very few people who are very, very, very tall, but we have some people who are kind of short. We have more people who are middle-ish. We have a lot of people with an average height. We have some people who are pretty tall, some people who are really tall, and then just a very few people who are super, super crazy tall, like 7'11" tall. Generally, there is no distinct category, so we can't say you are short or you are tall. We have like super short and a little short and somewhere in the middle, and so no really distinct categories. Tends to be more quantitative, so we're saying you are 5 foot tall, you're 5'6", you're 6 foot tall, controlled by lots of genes. We're going to learn on the next slide. This vocabulary, polygenic inheritance, literally means many genes contribute to this trait, also tend to be super strongly influenced by the environment. Human height is not just a product of your genetics, but also is a product of your nutrition and your exercise regimen and lots of other things that are happening to you. That's different from discontinuous or discrete variation, where we have very distinct categories. An example of this is blood type. So you can have blood type AB, you can have blood type O, you can have blood type A. I know this is not a good example. I need one more column. Blood type B. Tends to be more qualitative. I'm not saying that your blood type is 5 or 7, it's AB. Just a few genes or maybe even one gene, and tends to not be affected by the environment. So discrete, very categorized. Continuous is more of a range of phenotypes. Another example of a trait with continuous variation is eye color. So eye color is definitely polygenic. It is coded for by many, many genes. Sometimes we get super simplified and we're like, brown is dominant and blue is recessive, but there are so many amazing colors in between brown and blue. There are at least 8 different genes that help us get our unique eye colors. Some of them code for how much melanin you have in your eyeball. How dense is that melanin? Is the melanin more toward the front of the iris or more toward the back? What's pretty cool, that melanin pigment molecule, pretty much the same in our eyeballs. All different kinds of eyeballs have that same pigment molecule. It's just a matter of how much and where is it. And so again, eye color, definitely not brown versus blue, right? So many amazing colors in between. And it is a polygenic trait coded for by many more than one genes. One of the ways that we can analyze our polygenic inheritance phenotypes, those that show continuous variation, is by using super amazing box and whisker plots. I know that you learned about these in math class, so I'm not going to spend a huge amount of time talking about them, but we can put the median here in the middle, and then we're going to put the 25% of the data points that are under the median between the median and that first lower quartile, Q1, in the box. The 25% of the data points that are between the median and the upper quartile, Q3, are going to go also in the box. And then we go into the whiskers, where we have that lowest 25% of the data points and the highest 25% of the data points are going to go in the whiskers. Here is a lovely box and whisker plot showing some human heights. Here are those heights. We have males, we have females. Here are our medians. Sometimes we also throw some super weird outliers outside of the whiskers. Super interesting. And on that note, my friends, we have arrived at the end of our video lecture. We talked about pedigrees. We looked at autosomal, not on the sex chromosomes, dominant in every generation, recessive. We'll skip generations, but we'll have equalish numbers of males and females. Incomplete dominance, we can show that in a pedigree with some intermediate colors, gray instead of just blacks and whites. Sex-linked traits, we can show that in a pedigree. Female carriers will have half a circle colored. Males are hemizygous, and so more males will be affected than females if we're looking at a sex-linked trait. We talked about variation can be continuous or discrete. Polygenic, literally many genes, and then box and whisker plots can be used to evaluate that continuous variation. Great work today.